Hi, James.
Hi. Is it better?
Yeah, so it's coming from your end, and I'm wondering if you have an extra set of speakers that might be close to your mic.
I don't actually have any speakers at all.
Are you getting any feedback on your end?
No. I get a little bit of a squeaky noise, like an artifact, just a electrical artifact, not an echo. Could it be that my voice is reflecting off of the monitors in front of me?
It's squeaky when you talk?
No, only when you talk.
Okay. Maybe that's just me. So what I'm getting is my voice coming back at me about a second or so after I speak. And I think it's coming from your end, but the way we can deal with that is to just have you mute your mic when Erlen is talking, and then unmute it when you're answering the question, and that'll be perfectly fine.
So maybe what I can try is to lower the sound. With my speakers, I'll drop my speakers down. Try it now.
I'm also getting a delay. Are you seeing that?
Delay.
Problem is I can't hear you. So.
That's true for now as well.
Okay.
But that it wasn't distracting in the video.
Okay. So do you see where the mute for the mic is when you hover over the top?
I was actually looking at. Oh yeah, I see it up there.
So you can just toggle that on when Erlen is talking, and then just toggle it off when you're ready to answer.
Okay.
And that should do it. It won't pick up any of the echo on the recording, Erlen. It's just hard for you because you'll hear yourself back.
Oh, okay. Okay,
yep.
Good.
The recording will be fine.
Okay. Okay, that's fine.
Yep. Okay, I'm going to let you guys
Yeah.
get started.
Okay.
So when he's talking, I mute, and when I talk, I unmute. Okay.
Yeah, so when Erlen is talking, just mute that mic.
Hello, Jim.
Hello, Jim. Yes, hi.
Yeah, it's good to be here with you today. Here's how this will work. I'm going to adjust the camera a little bit for me. Great. And I have the questions that are scripted and that I sent to you. And I tell you, Jim, if you could, just while I'm talking, maybe try to mute the microphone. It is mute now. Okay, fantastic. Okay, great. So I have these scripted questions, and I will go through these questions. I'll try to be clear when I'm finished with the question. I will try not to drag out my sentences so you don't know when you're cutting me off or when I'm actually done. You can respond back. I'll try to keep an eye on the time so that we're not going over by too much. And I will control from my end how the camera runs. So I can control whether on the screen, whether the real video feed is showing you. So if you're talking, I'll switch it to you. If I'm talking, I'll make sure it's on me. Now, there's a good chance that I will mess something up during this interview. And so if that happens and I bobble over something, or if you feel uncomfortable and you want to stop, we can absolutely stop. And then we'll go back to the previous question and we'll start from there, because as we go back and forth, we can, those are good editing points that our videographer can work off of. So does that make sense? Jim, does that make sense?
It doesn't seem to be unmuting my.
We lost Jim. No?
No. It's a little bit slow to mute and unmute.
Okay,
I've got a
I'm going to turn up the volume. There's my delay.
Can you hear me now?
Tell you what, let's start this out and let's just see how it goes.
Okay, sounds good.
Okay. Okay. Great. Can you again? I'm going to introduce you as James Mangold and just refer to you as Jim for the rest of the time. Is that acceptable? And you are. I'm going to refer to you initially as a scientist at Novartis as well as a director of one of the programs at Novartis. That's fair?
Yeah, that's pretty accurate.
Okay. And one subtlety I need to ask about is Novartis versus NIBR. I don't fully appreciate what the difference is. Can I just say Novartis throughout the interview if I mention Novartis, or do I need to be careful and say specifically Novartis of Institutes of? Do I need to go through the whole description, or what do you think? And I think you're still muted. There we go.
The NIBR group is actually a division within Novartis. It really comprises the research division. So it's yeah, basically the people that I would say create value to the company.
Okay.
So, yeah. So I mean, you could just say Novartis in general, but acknowledging that the NIBR group is actually the research part of the group. We have divisions that do consumer health and all kinds of other things. So that's the distinction. It's really the research part of the organization.
Okay. If you feel that it's important for that to be clarified, I'm going to leave you to clarify that, okay? Because I think you're going to do a better job clarifying that rather than me. But the language in the course, in the online course, has all been approved by NIBR. And so I'm going to try just to steer clear of that distinction. Is that fair?
Yeah, I think that's okay. Probably, I mean, I can make a comment about it at the beginning. If you want to ask me, what is your current title or role, and I can say, I am, director in this department within the Novartis Institutes for Biomedical Research organization, which is the major research division of Novartis, something like that.
Perfect. Okay. I think that'll work great.
Yeah.
Okay,
occasionally you freeze up occasionally. So if I seem like I'm not doing something, it's because you've frozen for a second.
Okay. I'll try to be patient. This is not seamless, so we'll give it a whirl.
Okay.
And I'm going to practice switching between.
Are you still getting an echo?
Yes, I am picking up an echo. So I'm hearing my voice through your speakers delayed a little bit.
Okay, so I'll mute myself.
Well, ideally, I'm not going to be talking as much as you. When you're talking, it is absolutely clear. So I think my challenge is I need to be concise and then get out of your way so you can talk. So if you bobble muting, I wouldn't worry about it.
Okay.
Okay.
I'm concerned if I mute, the timing might get thrown off because there is a bit of a lag, as you said. So let's just try it and see what happens.
Sure.
He could go off. He could not mute the monitor and get used to hearing yourself and not worrying about it.
Yeah, Kristen makes a point that maybe I just need to deal with hearing myself, and that's fine by me. I can swing that. I hope. Okay. Well, right now the camera's on me, and I'm going to open this up. Start with the first question, and then we'll go to you for the next one.
Okay, I'm going to mute myself.
Okay. Yes, correct. Sure. Okay. Kristen is going to step out. And here we go. It's my pleasure to be speaking with Dr. James Mangold. Dr. Mangold is one of the research scientists and actually a program director at Novartis. So my first question to you, Jim, is what is your position at Novartis?
Well, currently, my group, I'm a director in the Global Drug Metabolism and Pharmacokinetics group, which is part of the Novartis Institute for Biomedical Research, which is the, I guess, the main research arm of Novartis Pharmaceuticals.
Perfect. Now, how did you come to this position within Novartis? What is your educational background, your research background?
Well, it's an interesting path that I have taken. I started off going to pharmacy school because my father was a pharmacist and my grandfather was a pharmacist. But in the process of going through pharmacy school, I had the opportunity, because I needed money, to work for one of the medicinal chemistry professors who needed someone to do some synthesis. And I got really fascinated in med chem. So to make a long story short, I went to, instead of becoming a licensed pharmacist, I went to graduate school in medicinal chemistry. And in the process of being there, I became interested in bioorganic chemistry and drug metabolism. And I guess, to make a long story short, I ultimately became a professor of medicinal chemistry for about 9 years, and then I saw an opportunity to go to industry with a lot of really interesting challenges. So that's when I joined essentially one of the legacy companies that's part of Novartis. So it's an interesting path. It started out in pharmacy school and ended up teaching, and then ultimately coming to a pharmaceutical company, and the challenges just don't stop.
Yeah, that's great to hear. Science does present challenges, and it's always stimulating. So could you describe some of the day-to-day activities that define your position as a director of drug metabolism and pharmacokinetics with Novartis?
Well, that's a question that to answer could take days. I think one of the things that people don't appreciate when you're working in a field like drug metabolism is how multidisciplinary it is. And I always tell people who are new to the company that you become the most valuable when you appreciate all of the disciplines that are part of drug discovery and drug development. The more you understand what's on everyone else's mind, the more valuable you become. So I think on a day-to-day basis, much of what we do is the discovery, mid-discovery, all the way through registration. And I think the type of questions that come up, the issues are very diverse. Many of them may be chemically oriented, they may be biochemically oriented. Much of what we do is multidisciplinary. So on a day-to-day basis, I could be working on a submission for a new drug, or I could be working with a medicinal chemist who's got a problem with metabolic hotspot he's trying to get rid of. So it's completely diverse in terms of the types of issues, yeah. So it's, as I said at the outset, it's always a challenge. It's always a challenge.
So I'm going to move this the interview more away from you and your experiences and talk more about your role and how it actually fits in with some of the content we've been learning in the course. So my first question to you reflects some of my biases that I bring to the course. I'm a synthetic chemist by training, and when I was first learning about drug discovery, I felt the challenge of drug discovery was let's develop a compound that binds as tightly to the target protein as possible. And then other minor details like pharmacokinetics and metabolism can be figured out at a later time, and then you'll have a new drug. Can you tell me why that thinking is so flawed for the modern drug discovery process?
Well, yeah, I think this is one of the having a drug that's developable, or some people like the word druggable, is really very, very appropriate. You can have the most potent compound in the world, but if it doesn't have the right pharmacokinetics, right delivery properties to its target, you're just simply not going to have something that's going to be viable. And I think that's important to understand early on, and I think that's why we invest so much time in trying to identify the molecules that are good actors and from ones that are not. Yeah, and I think again you can if, sometimes it's driven, I hate to say, from commercial standpoint. If you don't have a drug that's once a day, it just may be difficult to market because of the competition, or you may have difficulty with compliance. The whole idea is to make sure you make a drug that the patient can take it on a reasonable schedule, and not have to be concerned with such things. So again, having molecules that are potent is great, but you want to have a nice balance with the appropriate PK properties.
So as your group interacts with medicinal chemists, presumably people are making compounds and submitting them to some of your staff members for testing. What are some of the things that medicinal chemists, synthetic chemists, want to hear back from your group that are what would be a positive report from your group? What would characterize a negative report from those in your group?
Well, I think the first thing you want to look for is just a very fundamental metabolic stability issue. You want to know whether the drug is breaking down at a reasonable rate. And again, reasonable is defined by the indication and the dosing frequency. But in general, you're looking for a certain in vitro clearance that would be deemed acceptable. And again, you have to decide, what your pharmacokinetic and pharmacodynamic is. And I think that's another thing that sometimes people just think the only thing that's important is pharmacokinetics. It's actually the pharmacodynamics that is actually more important, because you want to make sure that you have the desired effect for the longest period of time, and many drugs behave differently depending on their target. You may be able to hit the target and you have a very prolonged effect depending on the nature of the molecule. So understanding the biology is important, as well as the the fundamental aspects of potency and interaction with the target.
So what sort of information would really be disruptive for the synthetic chemist? What would be bad news from your group?
Well, if we put the drug into our test systems and it disappears within 2 minutes, 5 minutes, generally that's a bad thing. But beyond that, there's other things that we like to avoid. They're not necessarily showstoppers, but we like to avoid any indication of reactivity. That is, many of our test systems, we include, for example, glutathione, which is an endogenous nucleophile that if we see an awful lot of the drug ending up as a glutathione-derived conjugate, that's not really a good thing. It's not a showstopper, like I said, but it's generally not a good thing. Small amounts, okay. Almost all drugs, if you test them, they will show some form of reactive intermediate produced, in particular, in an in vitro system. But generally, we prefer not to see that, or at least not to see it at a very extensive amount. So those would be what I would consider one red flag. The other red flag is we don't like to see wild differences between species. You don't want to see, it's completely different in human than any other of the safety species that might be included in the development package. Because that will be problematic in terms of trying to anticipate the human risk. So those are the kinds of things that we would consider maybe yellow flags, sometimes red flags.
Great, that's very helpful. So your group also, I believe, works with drug-drug interactions. So could you describe the type of tests or screens that you use to identify a drug-drug interaction?
Okay, again, that's a topic that could take days to describe, but basically, if you think about any particular disease you're going to treat, you have to appreciate that the patient is also often going to be on co-medications. If you come up with a very promising new drug molecule that interferes with the other co-medications, or vice versa, obviously this is a showstopper. A prime example was when one of the statins came out, and it had a particular sensitivity to a drug interaction, and even though it was identified early, it was put in the labeling, people still would inadvertently take the co-medication that would cause the adverse effect. It was so bad they had to pull it off the market because even though it was a well understood interaction potential, it still made the drug just simply too risky to use. So to get back to your original question, whenever we're studying the metabolism of a compound, we look at two things. We look at your drug as a potential perpetrator that is capable of causing interaction with another drug, or as a victim, that is, some other molecule co-medication could actually interfere with your drug's pharmacokinetic behavior and clearance. So in order to ascertain this, we first test the molecule for its ability to interfere with key metabolic enzymes or transporters. And this is done it starts at a fairly high level on a screen, and then it drills down to, I would call it the molecular mechanistic level, where we can kinetically define the nature of the interaction. If it's an inhibition, it's a KI determination. If it's time-dependent inhibition, which is a reactivity-based phenomenon, you will get this KI, K-enact, inactivation. So we get those kinetic descriptors from the ability of your drug to be a perpetrator. We'll also do the same thing with transporters. Can your drug interfere with transporters that might be important for the statins, OATP, ones like that? Now, from the other standpoint, to understand the potential of your drug to be a victim, you have to know an awful lot about how it's handled in the body. This involves what enzymes metabolize it, which ones are the most important, what are the major clearance pathways, renal versus metabolic or metabolism-based. So again, we define that through a series of tests. We use whole cell systems like hepatocytes for the overview. We use in vitro systems to define the enzyme kinetics involved, and we basically construct a model that allows us to fully describe all of the important clearance mechanisms for the drug. And all of these ultimately get fed into more comprehensive mechanistic models that allow us to essentially predict. And the tools have become extremely advanced in this area, and it's become a fairly large amount of what we do is taking the laboratory and using to build mathematical models that allow us to predict clinical outcomes. We can use it for, again, drug-drug interaction predictions, for design of drug interaction trials, anticipating what the changes will be. So it's really become quite a mathematical extension of what we see in the laboratory.
Now, you touched on the topic of drug-drug interaction trials. Can you share when would a trial be warranted or not warranted? I'm sure there are many drug-drug interactions that don't warrant a full clinical trial. So can you share a little bit on that process? James.
Okay, much of the decision process about when to do a trial, a drug-drug interaction trial, is partly driven by safety. If, for example, if you know you might have some sort of a safety concern, if you have a drug interaction, you might want to position it early. If you're going into an indication where you know everyone is going to be on, for example, a statin, you may want to position a statin drug-drug interaction trial very, very early, because this could be, again, a showstopper for your molecule. But beyond that, I think often what we do is we ask the question of, first of all, how big of an impact will the interaction be? And again, this is where your modeling and simulation efforts help you to know, is it going to be a very strong interaction, a weak interaction, moderate interaction? And this will then allow you to judge, if we think it might be strong, maybe we should try to do it earlier. So really a lot of it, the timing is based around all these kinds of very case-by-case project decisions, but a lot of it's determined by safety and the viability for the molecule. If it turns out to have an interaction, do we have to do a dose adjustment? Maybe such as maybe anti-HIV drugs, you may be able to use the drug interaction to your advantage in a combo therapy. And as you probably know, Novartis is very much into oncology indications, and almost all of the patients are on co-meds, and so you have to constantly be worried about interactions and how to anticipate them, and maybe even, as I said, use them to your advantage.
Jim, I'll ask one last quick question, and that is, from your language, it seems that the decision on what type of trials to develop, that is primarily driven by the developer of the drug. Does the FDA, would the FDA ever, in consultation with the FDA, would they ever ask, well, why don't you do a trial for this aspect, or is that really driven by the developer of the drug?
Well, I think the developer of the drug obviously shares a lot of the responsibility, but very much of what we do is driven by the regulatory expectations. For example, in terms of drug-drug interaction activities that we do, there are fairly specific guidances provided by both the FDA and the European health authorities, which specify, or at least make it clear, what their expectations are as to how you will investigate drug-drug interaction potential. Some of these guidances are very specific, and so what we try to do, and we try to keep not only up to date, but at least at Novartis, we try to be slightly ahead of the game in terms of having in place the tools to be able to get the information that we need to make the health authorities more pleased with our packages. But that being said, a lot of what we do also is based on our desire to make sure that we have the best molecules. We don't want to put any patients at risk. And toward that end, we feel a very strong responsibility to make sure that we really know what's going on, and we don't have, I guess, we don't like surprises. Yeah, I think a lot of what we do is driven by regulatory expectations, and we have no qualms about interacting with the health authorities, either to share our thoughts, what do we think will work well? Maybe we have a plan and we'd like their advice as to whether they think that's appropriate. And so I think there's a fairly significant dialogue between the various pharmaceutical companies and the health authorities. There are shared scientific interactions to try to come up with sort of a consensus of what's the best way to solve the problems that all of us have a stake in.
Jim, that's fantastic. I want to thank you on behalf of DavidsonX and also the edX course for making yourself available for this interview. And it was a pleasure interacting with you and having your questions contribute to the course.
Okay, thanks. I really appreciate the opportunity.
Take care. Take care.
Okay.
Hey, I think that'll work.
But you're supposed to go like this. Take.
Yeah, I've seen that a bunch. I have seen that a whole lot. Well, one thing I enjoy most about the discussions is hearing the language that people use to communicate in their field. So to hear, this is a showstopper. That's not a term I've heard. I've heard deal breaker a lot, but showstopper, the victim and perpetrator, just the different descriptive terms that people come up with to communicate in their field. So I appreciate your sharing all that, the terminology and your answers.
Actually, if you look at the terms, actually those are common in the field. Did you catch that part?
That's great stuff. Yeah, I did. You said it's in. Some of it dropped out, but the guidance is use those terms.
Yeah, you know, I actually sometimes when I speak to people, I have to be careful that I don't use too much jargon and too much of the yeah, the language that you commonly use. If you're working with a medicinal chemist, you talk about brick dust, and you talk about
Yes.
things like that. You know, everyone knows what that means, but people who are outside people, they look at you sort of funny, like brick dust. What are you talking? Oh, I know what you mean. So it's really quite a yeah. But I think what people really don't realize is how much interaction there is between, for example, health authorities, scientists, and industry people. We have there's a group that used to be called PhRMA, which is now called the IQ Consortium, which is basically a bunch of people who from the industrial side, also a few people from the academic side. They get together and they discuss issues around drug development and the like, and we try to come up with white papers, you know, consensus, and then the FDA will come out with their response, and then you would meet some common ground, and there'll be even meetings where, you know, industrial and FDA people will actually discuss, you know, what makes sense, what doesn't make sense, because, you know, we're all in this together, and we have to figure out, you know, how to leverage the science we work with. Everyone gains then.
That's great stuff. Well, Jim, I don't want to take up too much of your time because I've already overstepped what I committed in the email. But it was a pleasure talking with you, and thank you again, because this is, you know, it's one thing for an academic to present the material in the course, but to hear a professional in the course and use the language of their trade, I mean, that's an important addition to the course. So, you know, people like you and others in Novartis are helping that out.
Yeah, I think having been on both sides of the podium, if you will, I've stood up in front of pharmacy students and tried to explain them what drug design was, and a lot of them glaze over, and some of them say, "Whoa, that's cool." So, and then becoming the guy that actually does this kind of thing, it really gives you I would say it's humbling in some ways, you know, because being an academic, you think, oh, you know, it's all just rosy, but actually it's really tough. It's really tough to come up with a good molecule. It's got to make it all the way to the clinic. And that's very, very hard to do. We get a lot of molecules that look really, really great, and then they hit phase 2, and what happened? You know, it's just, you know, they're either too variable, or maybe the efficacy is just not good enough, not quite good enough. So it's a tough business.
Yeah, no, well, you know, it takes skilled people, and I'm glad you're one of those skilled people. Well, I'll let you go, Jim. Again, it's a pleasure, and if you have any questions, please don't hesitate to send me an email. But this video will be edited, and then we'll end up posting it within the course.
Okay, sounds good.
Okay. Take care, Jim.
Okay, take care.
Bye-bye.
Bye-bye.